Good morning. Oh, whoops. Some kind of camera problem. There we go. If you are here, please let me know in the chat. It looks like almost everybody did that already. Oh boy. Right, exactly, Caitlin. So if you go back and watch Friday's video, you can just skip around, but I go through it like step by step what you need to include in the project. I think it's at the beginning, and Friday's pretty short because the lessons were pre-recorded, so there's not like a ton of stuff for you to skim through. And I also cut out all the empty space, so it'll be a fairly short video, and you can just skip around and find it. But it's definitely there. Do you do an entire family or just one species? Both are acceptable. So you can pick a species. In which case, just make it clear that you are doing this species, use the correct scientific name, or you can pick a family. For example, I mentioned that maybe you want to do bamboo, but you don't want to do like a bamboo species. You just want to talk about the general purpose, like general uses of bamboo as like a food source for bamboo shoots and heart of palm, or I guess heart of palm is from some different plant, but used as a water source, used as a building material. You could just do the family of bamboo. So you don't necessarily have to do bamboo as a like one specific species of bamboo. It's a little bit too restrictive, especially for certain types of plants. Like bamboo is a great example because when people say that they like built their lawn furniture out of bamboo or like their sheets are made out of bamboo, they're not made out of like a specific species. I mean, they are made out of a specific species, but not every bamboo pair of sheets is going to be made out of the same species. So in other words, like you need to talk about bamboo more broadly than just one species. No, Kim, I don't think that's nowhere in the instructions. You don't need to go through the full taxonomy, but you do need to either tell me the species name if it's a species, like the full binomial nomenclature, or if it's a family, you're going to give me the family name, but at least tell me that it is a family, just so I'm aware of what specifically that you're talking about. Family blank. Can we take up the analysis questions from Friday, specifically STEM 2 and Roots 1A? Yes, absolutely we can. Let me go find those. Stems 2. Okay. Examine figure 2, cross-section of sugarcane on page 577. Let me pull my textbook out here. Let me just take a quick photo of this image here, and then I can just paste it into my document here. So that's the photo. So it says, is sugarcane a monocot or a dicot? In which cell types do you predict that sugar is stored in these systems? Or in these stems, rather. Okay, so first of all, monocot or dicot? You can't see it. Thanks for mentioning. This could have gone on for a while. Sorry, give me one sec, guys. I've put my laptop here on a network cable, so we should have a lot fewer connection problems than we've been having previously. I'm hoping none, but we'll see. It should be a lot faster though. I'm basically off the house Wi-Fi. Okay, there we go. So this is the picture from the textbook. So let's start with this then. Monocot or Dicot? Anyone want to take a stab at it? And if you are choosing Monocot or Dicot, brief explanation as to why you've chosen one or the other. Okay, good guess, Michaela. Do you want to take a shot at an explanation? Why are you saying you're right? By the way, it is a monocot. How do you know it's a monocot? Ah, there you go. Lillian beat you to it. Sorry, Michaela. So you've got distributed vascular bundles, and you can see them there. There's xylem and phloem tissue distributed. The big empty ones are the xylem for transporting water, and then the small cells that are adjacent to them towards they're yeah, here. Yeah, the arrangement of them is a little bit unusual here. Usually the xylem are found towards the inside. The arrangement's a little bit strange here, but the smaller cells that are distributed around, they're actually oh yeah, I can circle on here. So these are the phloem, or sorry, the xylem. Rather, we better not mix those up. For transporting water, and then always towards the outside are the living cells, the phloem cells. So the phloem are actually this little grouping of cells right here. Some of these are oriented a little bit unusually. I mean, it's a biological system, so it's not going to be perfect, but usually towards the center is always where you find the xylem, and the phloem are more towards the outside. So just to pick another one here, we've got these right here, which are xylem, and then the phloem are the cells towards the outside. These are living cells, the phloem, pumping things, whereas the xylem are basically empty. They're acting like straws. Okay, so question. Sure. So Kim, good. So what you're saying is vacuoles within the cell. That is where within a cell one would store water-soluble content like sugars. So yes, within the cell. But I think this is asking which type of cells. Let me reread it here. So first of all, we got monocot. In which type of cells do you think that the sugar is stored? So going back to the type of cells. Let's go up to monocot distribution here. So there are three basic types of cells, tissues, in a monocot stem. You've got your epidermal tissue, which is around the outside. You've got the meat, which is basically just the cells that are distributed throughout the stem. That's the ground tissue. And then you have your vascular bundles that are coming up. So the vascular bundles are not storage cells. They are transport tubes. The epidermis is protective covering on the outside. It would be what's forming the bark of a dicot. There's no bark on monocots, but it would be like that. So there's really only one option here, which is that it's going to be found in the ground tissue. There are different types of ground tissue. We didn't get into a ton of detail on the different types of ground tissue. I don't think. Did we talk about parenchyma, sclerenchyma? No, I don't even think that's in this note. That's from a different course. I don't want to get into too much detail here about it either, because it's just not stuff you need to know for this course. So So it's basically ground tissue is what you need to know. The structural tissue of the center of a monocot stem. There's another word for the ground tissue in monocots. It's called pith. So I guess I could write that right in there. It's called pith. And in sugarcane, that's where you would find the main storage of sugar. Now, there's other places where monocots store sugars. You could store them in the roots. You could store them in sort of some pseudo root-like structures, stem extensions like potatoes do. Because the potatoes themselves are not actually roots. I guess they're really more like an underground extension of the stem. But anyway, different plants have different strategies for storage, but the sugarcane specifically, if you guys have ever seen somebody harvest sugarcane and like process sugarcane, you basically have to like press the stems, and when you press the stems, that's what the sugar comes out of it. So hopefully that answered the question. I think it did. And then the second question was stem 2 and roots 1A. Roots 1A. Epiphytes. So this is just a question that requires research. Are epiphytic plants common? They're actually surprisingly are common. You can find them in most terrestrial ecosystems that have dicots, which is most. You can find epiphytes. Epiphytes include things like holly, all kinds of different climbing vines. Although not every climbing vine is an epiphyte. Epiphytes specifically drill their root structures into another plant and kind of steal from the other plant. And so not every climbing plant is actually an epiphyte, but many of them are. Things like or some types of orchids are epiphytes. They actually grow into some dicots. Does that answer your question? I think that's the first part here. Are they common? Is just like basically a Google search. How many epiphytes are there? Are they relatively common? Yeah, they are. There's a whole bunch of different kinds. There's certain ecosystems where they're more common than others. You should look that up. I want you to look that up. Where do you find epiphytes? Okay, all right. Cool. Today, we are on day 17? Tomorrow's the last day of the unit, right? Yeah. So we're on day 17. Yes, and then the second half of the day is going to be our flower dissection. So, first half, I've got a really cool video here. If you're into physics, and some of you have got to be into physics, this is a Veritasium video. I love this guy. He's got a lot of great content on YouTube, but this one specifically is about the physics of how you transport water up a tree stem, and like why the things that trees do with regards to transporting water are like really amazing, like truly amazing, that a tree is able to pull water up 100 meters of stem. Because if you were to try and use a pump and like put it 115 meters up, it's not actually physically possible to pump water up 115 meters, or I should say pull water up 115. And keep in mind, and as you go through the note for this, you'll see that water is largely being pulled up the stem from the top. So if you took a pump, and this could be like the greatest pump of all time, it could be a what are those vacuum pumps called? Is it micro vacuum? Anyway, the pumps that you use to get like the lowest possible vacuum, the best possible vacuum. And by vacuum, I mean completely remove the air from something. If you were to put a pump like that at the top that is capable of getting to virtually zero pressure, okay, you still would not be able to pull water up a length of pipe that long. Doesn't matter how small the pipe is. And then this video talks a little bit about the physics of that. And if you're really interested in the physics of that specifically, there's a whole separate Veritasium video going into why that is. But they talk about it a little bit in this video. So there are some other really cool things that trees are able to do to make this physically possible, to actually pull water up to the top of a large structure. And so this is going to go into that. This is mostly about water transport. We're going to talk about both. So we're going to talk about the three mechanisms that plants have for transporting water that we know of anyway. And then we're also going to talk about how plants transport sugar. Okay, so obviously the difference here is the water component is all about xylem, how water moves through xylem. And then the other component, the sugar component, is how sugar and nutrients are moved through the phloem. So, like I said, this is sort of an introduction to the physics of those mechanisms that trees use for water transport. And then in the note, we're going to go through both. We're going to talk a little bit about the anatomy of xylem cells and phloem cells. Those cells have specific names that you should know. And they have totally different strategies for moving stuff around. And the reason why xylem and phloem are so different is that one is basically unidirectional. There's only one direction you ever need to move water in a plant. You need to move it from the root to the leaf. That's so that the water can get used for photosynthesis at the leaf. So it's exclusively a one-way system. And so it works completely differently than the sugar transport mechanism in the phloem, because you need to be able to transport sugar wherever you need it. Are you building new buds in the spring? You got to transport sugar to the leaf. Do you have fully grown leaves and you need to transport the sugar that it's making to a storage area? Well, you got to transport in the opposite direction, from the leaf to the root or to the stem or wherever that particular plant keeps its sugars. So anyway, they're completely different systems. They run parallel to each other, and they interact with each other, but they are doing two completely separate tasks within the tree. Or I shouldn't say tree. All plants that are vascular have this, but we're mostly going to be focusing here on monocots and dicots. And in fact, a lot of this video is talking about dicots because it's referencing trees all the time, and trees are almost exclusively dicots. So anyway, you're going to run through this lesson. It's going to go through the different mechanisms and the anatomy of the transport system of the plant. There are 4 questions from the text, and then there's a little exit quiz that you do at the end. Okay, this lesson's a little bit longer. It's 42 minutes. But I don't think this will take you the whole first block, but who knows? It may. This is also chapter 12.5 in the textbook, and so again, you can feel free to reference chapter 12.5. If you ever want to like read the chapters that we're doing, you're welcome to. A lot of the information that is in the notes comes directly from the textbook, although I mean, I gather information from all different sources, but a lot of it is covered in the text as well. So if you're looking sort of for a refresher, you're always welcome to read. Some people do better when they're reading. So then when we come back from the break, we're going to do a flower dissection. So yesterday, hold on one sec. I took a walk through the forest and I found a number of different wildflowers. Unfortunately, I didn't find anything very large, which would have been very handy for dissection purposes. These are mostly pretty tiny. I borrowed a digital microscope from the school So I'm going to try and get that set up and running. Unfortunately, it's actually fairly old. You wouldn't think a digital microscope would be old, but I think it's from the 90s. So I don't know if it'll run on Windows 10. I'm going to play around with it and we'll see if we can get her going. But anyway, I'm going to look at some of these flowers with you. I'm going to cut them up and let you look at the different parts, the male and female parts of the flower. We're basically going to do a flower dissection. If you have your own flower, I'm going to ask that you try and find those as well. Oh, these actually smell really nice. That's nice. I couldn't smell them yesterday. So anyway, we're going to do a flower dissection. We are, there's a little reading that goes along with that. And then there is an awesome documentary called The Power of Flowers. It talks about the evolution of the flower, why they exist, why the traits in the flowers evolved in the first place. I can't recommend this documentary enough. It's fantastic. I'm hoping this link still works. It keeps breaking because I don't know how legal these links are in terms of copyright. I mean, it's on this website, so maybe it's fine. I don't know. I'm not sure what the status is of this, but anyway, I'm not hosting it, so there are some. I believe that there are some subtitles in a different language. You can learn a new language while you're watching it. There are a couple questions that go along with this, and then there's a short exit quiz at the end. Today we're going to talk about flowers, and tomorrow the last thing we're going to cover is we're going to talk in more detail about the sexual and asexual reproduction of plants. So how do plants reproduce? And then we're going to get the second half is for our assignment. Okay, guys, I'm going to let you work. Please let me know if you need anything. Hey guys, welcome back. If you haven't already done so, let me know in the comments that you are here for the second half. Okay, so for the second half, if you have a flower, that would be very handy. I found the flowers that I'm going to use at or in the forest behind my house. So what we're gonna do, it would be helpful. Oh, I got you, Ryan. If you had the worksheet out. Let's see. Oh, right. Sorry, somebody asked me to answer a question. The part B of question 4. How would this affect transport in the phloem? Okay, so this is actually kind of a complex question because what you're doing. Let me draw this on here. And I realize I got to put my screen on. Okay, so imagine you've got a continuous xylem. We're talking about blocking the stomata. Okay, so the stomata are at the bottom of the leaf. So you got your leaf. I'm gonna just do like a side view of the leaf. Okay, so there's the bottom epidural layer of the leaf, the lower epidermis, and it's got these stomata cells with a gap. Okay, so there's gaps here. And then there'll be, sorry, I was intentionally trying to draw these squares because they're typical plant cells, more geometric, square, prism, cube ish. And then the stomata cells, the guard cells rather, are more of a semicircular shape. Okay, so basically what they're saying is what would happen if instead of having these gaps in the bottom of the leaf, you just like nail polished over them and blocked all the gaps off. Okay, so you have to think about what effect that's going to have on the plant. So what do the gaps do? What do the holes in the leaf do? They are for exchange. So they allow moisture to come out, okay, because remember that there is water being used for photosynthesis. However, a lot more is being transported up than is being used for photosynthesis. So there's a whole bunch of, if I could draw the xylem and phloem up here. I mean, I'm not doing these justice because I'm kind of rushing, but okay, imagine that this is the xylem, and then you've got your phloem next to it. Obviously the colors here are kind of made up. So the xylem is the one that's playing the active role here in photosynthesis in terms of bringing water up. And so there are these little micro passages here. And as water is being drawn in, I probably should have drawn this a little bit bigger, but I'll zoom in. So we've got little H2Os here. As H2O is making its way out here, it's actually drawing due to the stickiness of water, the molecular stickiness of water called surface tension. Sorry, that took me a second. Through surface tension, it's pulling additional H2Os out, and these H2Os are actually evaporating. They're in a gaseous form here. Let's make a copy of this. There's a bunch of them in here. And so as O2, H2O, as water is normally drawn out of here, more H2O is going to be pulled through these little vents. Okay, that's called transpiration. They talked about that in the video. Okay, so as O2 is leaving the leaf, or I keep saying O2, H2O is leaving the leaf, it's going to draw additional H2O out into the gases in the spongy mesophyll here, which is going to pull additional H2O up and out of the pores in the xylem tissue. Okay, so that evaporation is one of the, they're called transpiration or evapotranspiration, is one of the main mechanisms that keeps this continuous pull of water molecules coming out through these little pores from the xylem into the tissue here. And remember, they need to come in so that the, I mean, I'm not drawing all of the cells that are here, but there is the palisade mesophyll, which is right here, which needs those water molecules, right? So they're actually going to be pumped in by these palisade mesophyll to be used as part of photosynthesis. So it needs the H2O for that purpose. And this continuous cycling where they're exiting causes more H2O to get pulled out through the pores and into the spongy mesophyll. So if you seal this off so that there's no gas exchange, well, you've stopped the evapotranspiration from occurring, which is effectively going to completely halt water transport through here, through the xylem tissue. So that's what happens to the xylem, and I think that was the first question. Predict how this will affect water transport. It basically stifles water transport if you close off the escape for water. You're also stopping photosynthesis from happening because you're not allowing the ingredients for photosynthesis, which is CO2. You're not allowing CO2 to come into the leaf to be used for photosynthesis, and you're also getting a buildup inside here of oxygen. So you've got O2 that's being produced by photosynthesis, and it's going to build up in here because it can't vent out into the environment. So all of those things are going to basically put back pressure on photosynthesis and prevent the photosynthesis chemical reaction from occurring. Okay, so you're going to put a halt on photosynthesis if you block the stomata, and you're also going to stop water flow. Now the second part of this question, the B part, is a lot more complicated because remember that the phloem is transporting, let's assume this is during the summer, okay? Well, the phloem is going to be transporting the sugars from here, from the leaf, down the plant to wherever it's going to be stored. Maybe it's in the roots, or I don't know. Okay, it depends on the type of plant where you're going to store this, but let's say that it's you're trying to get it to some root cells, which are down here. Okay, and there are these companion cells, and then there might be some larger cells down here that are used for storage in the root. Okay, and then this cell right here would be the companion cell. Okay, and then there's also companion cells on this end. They're always sitting adjacent to. I'm really not doing a great job of drawing these, but like let's say this is a companion cell. Okay, right here. So what we want to do is we want to move sugar. I'll just draw a little sugar molecule here. Okay, there's our little sugar molecule. We want to transport this sugar from here, which is where it's being produced at the palisade, into the phloem, and then it's going to get exchanged from phloem cell to phloem cell. Remember, the phloem cells are living cells, so there's multiple living cells here, and they're actually going to get pumped to some extent through. Okay, so this actually requires energy, and you're going to be transporting it from this cell, and then from this cell to this cell transported it, and then from this cell to this cell transported it, and from this cell transported it. There's some energy being used to actually transmit this sugar molecule all the way down through the whole chain till it gets to how did we refer to this in the note? We have the source and the sink. There we go. I had to think about that for a second. So this is the companion cell is going to pull it out at the bottom here and then potentially transport it into a storage cell down here where it's going to be converted into starch for most plants. Okay, so in some ways, if you block off the leaf, that really doesn't affect this process at all, because these cells are still going to be pumping sugar to their destination, except there is a little bit of assistance that the xylem provides in order to get this to work more efficiently. Okay, so it still works. You can still pump the sugar from the source to the sink. However, let me explain how the xylem actually assists with this. Okay. So we've got our xylem, and the xylem, remember, is running directly adjacent here to the phloem cells for the most part. Okay. And we saw that when we looked at our stems. The xylem and the phloem tubes are actually like right pretty much next to each other. All right. So if you look at the concentrations here, what you find is that there is more sugar here than there is here. Okay? There's more sugar in the phloem than there is in the xylem. And that should make sense, right? You've been pumping the sugar into the phloem, so there's going to be a lot of sugar there. Now, sugar cannot get across this barrier right here. This barrier is impermeable to sugar, so you're not going to get an exchange of sugar into the xylem tissue. It can't be transported across. However, it is partially permeable to water. So what ends up happening is that the concentration of water is actually lower here than it is here, because there's more solute dissolved stuff over here and less dissolved stuff over here. So another way to think of that is you could say that the concentration of water is actually higher concentration of water here than concentration of water here. Or you could say that the liquid in here is maybe 30% water. It's probably closer to 70. Whereas on the other side, it's like 100% water. In other words, the concentration of water is higher in the xylem. Okay, I talked about this in the lesson a little bit, but this is a little bit confusing. So what happens when you have a high concentration area and a low concentration area is you always get movement from high to low. That's diffusion, right? When it's happening with water, when water goes from a high concentration of water to a low concentration of water, we call that osmosis. But it's the same thing. Osmosis is just diffusion of water. Okay, so what happens because there's this imbalance of concentrations? You're going to get movement of water at this end. Okay, so water is going to move here. Now when you pump water into something, that creates a pressure. Okay? That's turgor pressure. All right? In plants, it's called turgor pressure when they have water pressure inside of them. All right? So what's happening here is that we have high pressure. This area here is high pressure of water. At the bottom, way down here, because this cell here at the bottom is removing sugars, remember it's pulling the sugars out, okay? So the sugars down at the bottom here are getting pulled out of this, which means that there's going to be a very low concentration of sugar here. This is low sugar concentration. If the concentration of sugar is low, that means that the concentration of water is high. Okay? So here we have high water pressure, or concentration of water. Okay, I'll use the concentration bar here, compared to what's in the xylem over here. So this is comparatively low water concentration. So what happens at the other end is the opposite, which is water is getting through osmosis getting moved into the phloem, or into the xylem rather. Sorry, into the xylem. And that causes a low pressure area down here, low turgor pressure. So if you have a low pressure area down here and a high pressure area here, then you're gonna get assistance, help, moving things from the high pressure to the low pressure. Okay, so that assists. That process of having this pressure differential across the phloem helps push sugar to wherever it's supposed to go. Okay, and that's assisted by the xylem. It only works if the xylem is continuously moving upwards. And so if you stop moving water through the xylem, you stop that process. And so this assistance that you get with the pressure kind of goes away a little bit, and all you have is just pumping of sugar through the phloem. Does it still move sugar? Yes, but it just moves the sugar slower. So there's a really, by the way, super long-winded answer to that question. That's not an easy question to answer, but that's the answer to 4B. So how does it affect transport in the phloem? It slows it down. It's not as efficient because we lose this pressure system thing that's going on that's helping move stuff through the phloem. But it still pumps it because it's an active process. It requires energy, so the pumps are still going to keep pumping. Did that answer your question, Luca? That is the answer to the question, but it's a roundabout answer. It's not as straightforward as the other one, because the other one is like, okay, well, if you stop letting water escape at the top, well, then no more water is going to go up the tube. That's pretty obvious, and then the xylem doesn't do anything. But the phloem is just a little bit more complicated. Okay, all right, cool. So what we're going to do in the second half here, let me pull up the website. We're going to take a look at a flower. So I have a microscope. Like I said, I borrowed, and I did get all the software going. Very exciting. I've never had this going before. I should start using this in the classroom all the time. To be perfectly honest, I didn't know that we had this until recently. But now that I know that we have it, and I know how to run the software, I'm going to use it more often. But it looks like this. It's like a little microscope on a stand, and I've got it hooked up with the USB to my computer. And I've got a couple different lenses on it for doing different magnifications. And so this is just going to help us look at the structures of the flowers, because the main problem with this is that when we do this in class, we do it with lilies, which are gigantic. So if you have a gigantic flower, you can see all of these structures with the naked eye. They're very easy to see. But if you have a little teeny tiny flower, like most of the flowers that I just found, I'll give you an example. Bring one of these over here so you can see it. you have a little tiny, teeny tiny flower like this is a whole bunch of small flowers on here. It's probably not even going to go into focus. That's going to be a lot more difficult. So I'm going to kind of jump back and forth here between the microscope and this camera right here, which is unfortunately not great. And I'm going to try and see if we can find all of the important structures of the flower on a couple different types. I don't know what you have at home. If you have a nice big lily or something like that, these will be very easy for you to find. If you don't, if you have some little tiny flowers like the ones that I have here, if you have a magnifying glass at home, that would be very useful for you probably. Although you may not have that. I mean, to be perfectly honest, I don't own a magnifying glass, so it wouldn't be that strange if you didn't have one. But you can also look just very carefully. They can be seen with the naked eye, but they're just small. So it certainly isn't going to show up very well on this camera. So what you need is the flower dissection sort of worksheet in your course notes. I believe it's page 14. Where's the last course notes thing? It'll be on the previous day. Course package. Did we get paid? Maybe we got on day 15. There we go. It's this page right here. Okay, where it says flowers. And there's a nice big diagram there. And if you have a lily or something like that, it's basically this exact flower. Very easy to see the structures inside. So good on you if you've got one of these. If you have something smaller, we'll do our best and we'll see if we can identify what you got. And if you don't have anything at all, that's fine too. I'll do my best to show you on here. So let me get set up first with my little iPad stand. I wish my desk was a little bit bigger. It's pretty, my area for working is quite small. Gotta put a million cables running through here. The reason I want to use the iPad camera is just to give you sort of a macro view of it before we get the microscope out, so you can see it like the large structures a little bit better. There we go. What you're going to do, by the way, after we look through a couple flowers together, is you're just going to read through this background information about flowers. I may as well tell you that now. Oh, that's my calendar. And then you're going to read page 598 in the text and use it to fill in the information at the bottom of that page. So that's just to fill in this table right here. Then after that, you're going to watch this documentary. It's a fantastic documentary called The Power of Flowers, or How to Grow a Planet, The Power of Flowers. And then there are just two questions that I want you to think about while you're watching the documentary. So there are these two questions right here. And there are a couple questions to complete for homework from the textbook. It's page 602, 7 and 8, plus a little Lexa quiz. Okay, so that's what we're getting into. But I'm going to go through a couple flowers with you now so you can see what they look like. So how do I do this? Like this? Is this going to work? So I'm going to use this view to just be able to give you sort of a larger view of the flower. This is going to come into focus. Oh, perfect. And then I'm going to get the microscope view, which I have. This is going to take a little bit to get everything into proper focus because it's not. This is not that straightforward. But I'm going to switch to microscope view, which is right here. And then we're going to use that to get a real nice close macro view of the structures. Oh, this works really well. This is definitely going to take some time to get stuff into focus. This isn't like focusing on a microscope slide because like stuff is moving around as I'm doing this, which makes it a lot harder to get it into focus. And this is just on the low magnification. Wait till we turn the high one on, that makes it way harder. Really cool. You can actually see little grains of pollen on the outside of it. All right, anyway, let's start with the macro stuff that you can see. Just give me one sec, guys. So this is also in the textbook. What page is this in the textbook? Flowers. 598. Okay, so first of all, sepals at the base. Okay, so let me line up all the different flowers I've got here. So I've got this one. I've got this one. I've got this one. I've got buttercup. I wish I could tell you the names of all these. I don't know the names of all these. This one's called a buttercup. And this one is a daisy right here. This one I've got no idea, and I don't know what this one's called either. This one. Some of these, by the way, are multiple flowers. They're not just a single flower. This is a number of flowers on little stems, and so is the first one here. This is a bunch of flowers. There's four in a cluster on this one. I've got these ones. And I've got these ones that look kind of similar, but they're actually a different species. They look similar to that other one, but they're different. Okay, so what's going to be the easiest one to identify these with? You know what? I actually think the buttercup is probably the best example of a perfect flower. So I'm going to start with this one, and then we'll look to see if we can find the same structures on other stuff. If you look at the bottom, where's my Apple Pencil? I'm gonna use that to point at stuff. Oops. If you look at the bottom of this one, if you look at these little structures down here, sorry guys, where's my little camera? Right there. Is it gonna be able to focus on this? Yeah, these little structures at the bottom right here. Whoop, like that one that just fell off. Those are sepals. Sometimes they're green, sometimes they're the same color as the rest of the flower. They were a protective covering for the flower before the flower bloomed. Now they don't really have any function any longer, but they were originally a protective cover for the flower. So, and sometimes they're a different color. These ones are actually white with a little bit of brown on them, and then once they having some difficulty keeping this in view. Once the flower blooms, they turn yellow and kind of shrivel up and fall off. Not on all flowers, they don't all fall off. So on this one, for example, the sepals are actually still basically forming the base of the flower. So if you look underneath, this, you can see them. They are like the little, I don't know what to call them, teeth? This right here is sepal, right here. These structures right here are sepals. So on this particular flower, they kind of stay attached and kind of stay as part of the flower. Wow, why am I having so much difficulty with this? Is it going to get into focus? Too close, too far? I'm so sorry, guys. This is, I don't even know which, this is an iPad that has like a bunch of cameras on the bottom. I don't know which one is actually the right one. There we go. That's better. So there are the sepals there underneath. Okay. Then the receptacle part is just sort of where the flower is meeting the, oh, this one just lost a petal. I should have grabbed some tweezers. Oh, this one's losing all its petals. Poor Buttercup. It might make it easier for us to see what's inside. So, anyway, at the bottom, right here, right at the base. God, this is in terrible focus. I'm hoping I can do better with the microscope. Right at the bottom where I'm pointing here is a little area that's the receptacle. It's not very obvious on this flower. This one actually has a more obvious receptacle. If you look at the base of each of these flowers, you can see a little green part, like right there, and right there, and right there. Why is this in such terrible focus? Oh, there we go. That little green part at the base of the flower is the receptacle. Okay, so that's holding the flower on. And when the flower is actually, well, eventually it's going to turn into a fruit, which is going to contain a seed. Eventually, when it turns into a fruit and the seed is mature, the seeds are mature, the receptacle is actually going to let go, and that's when the seeds are going to begin their journey, which is the eventual goal here for all flowering plants, is that they're trying to get their seeds out into the natural environment. Sometimes animals are taking them. If they're in like an edible fruit, then an animal is going to be taking them on a journey. Okay, let's look at the internal structures of the flower. So if you look at the top, and this is what I'm going to be trying to see with the microscope, you can see some little yellow, on this guy, spines, I guess you'd call them. They're on the outside, and they have like a little sac at the top of each one. They're these guys. Oh God, it's in such terrible focus. It's focusing on my hand. I think this will be easier to see under the microscope. So no, I'm not going to focus. I think I'm too close. I have my iPad like not facing me, so it's like upside down and backwards, which is really throwing me off here in terms of getting this centered properly. Come on. There we go. Okay, so those structures around the outside there are together called the stamen, and the anther is sort of at the very top of it. That's the little sac at the top, and the filament is the sort of stringy part that's holding it up. And then in the middle, this particular flower, why am I struggling with this so much? This particular flower, come on, get in focus. Get in focus. Get in focus. Come on. No, don't focus on my thumbnail. Ugh. Sorry, guys. This is really. That part in the middle has got a whole bunch of carpels. And you can't really see them right now because they're too small. We'll look at them under the microscope. But at the very top of each carpel is going to be a stigma. It's a little sticky spot, and that's going to be able to bind with pollen. Underneath each one is going to be a style, which is basically a tube that goes down to an ovary at the bottom. And the ovary at the bottom sometimes contains many ovules, which are the eggs for the flowering plant. Or sometimes it just contains one ovule, and it all depends on the type of fruit that is going to be produced. So if you think of like a kiwi would have an ovary at the bottom that has a bunch of seeds in it, or a bunch of ovules which are going to become seeds. Each ovule becomes a separate seed. Whereas, I'm sorry, I realize this is in terrible focus. I can't, how do I make this focus? Focus on this thing. There. Whereas sometimes at the base of the, sometimes the ovary is going to contain only a single one, like an avocado would be a good example of that. So there'll be one single ovule inside the ovary. Another example of one where there's only one seed per ovary would be like a dandelion or something like that. I believe that these buttercups also only have one ovule per ovary. So let's take a closer look at this thing under the microscope so you can actually see. I'm going to show you try and show you the male parts first, and then I'm going to see if we can zoom in on the female parts. So we're going to try and see filament and anther first. And I'm going to try and show you what you might even be able to see pollen if we use the really high magnification lens. And then we'll see if we can find it on some of these other ones as well. Let me switch over my view to the microscope. All right, in we go. This is also going to take a little bit of finagling to get this into proper focus. Yep. Perfect. Kind of, sort of. There. Okay, so at the top of the screen, up here, we're just seeing the end of the filament. And then this part right here is the anther. And if you look really closely, you can see some little bumps, little tiny bumps on the surface. There's a pollen grain right there. And that is the actual pollen sitting on the surface. I'm going to try now and use a higher magnification lens on this thing. And see if I can actually give you a better view of the pollen. So we're going to try it. Because these are not flat, the things that we're viewing, especially that was 100 times magnification, by the way. It gets a lot more complicated once you start increasing the magnification. So we'll do our best. I've got a 400 times magnification one here. No guarantees on this actually working, but we'll. I did try it earlier. It's easier on some flowers than others. The daisy is the easiest one, I think, because it's the most flat. So, all right, here we go. This is 400 times magnification. Oh, we're close. Just gotta get it in focus. Oh, come on. Oh, you can see them. You can see them. Even me talking. It's interesting. Just me talking is vibrating the filaments and the anther and the filament enough that it's kind of shaking around on the screen. There's 2 pollen grains right there. Can you guys see that? So that's kind of cool. There's 2 obvious pollen grains sitting next to each other. So remember that those pollen grains are the gametes for this flower, for this plant. So those are basically sperm, plant sperm. And each of those pollen grains is going to contain 1N chromosomes in it. Any questions so far about what we've taken a look at here? This is the male part. I'm going to see if I can find these on other plants too, but I'm going to try and do the same thing now here for the female part. Let me know if any questions are popping into your head as we're doing this. I got to go down magnification to get figure out how to look at this. So the female parts are a little bit more difficult to focus on. I'm going to pull the petals off. Oh, I forgot to talk about the purpose of the petals. They are basically signposts designed for insects, for pollinators. They talk about that in the documentary quite a bit, actually. And they often have patterns on them that are not visible to the human eye, at least, because the patterns are... I'm going to try and remove some of these pollen sacs so we can see the female parts a little bit better. These are they're visible to insects, though, I was going to say, because they're visible under UV light, which humans are not able to perceive that part of the spectrum. Great. There's some great pictures of it in the documentary. So, okay, I removed some of the little pollen things here. Let's see if we can zoom in on this. We'll do our best. In we go. Zooming in. We'll get there. We'll get there. Looks like an alien world. Ah, perfect. All right, so there, what we're seeing is the female parts. Perfect, perfect, perfect. So here we have the top of the female part. The female part, by the way, is called a carpel again. So the top, the very top part here is the stigma. So we've got a stigma here, stigma here, stigma here, stigma here, stigma here. Okay, so that's the stigma. It's sticky because it's intended to stick to pollen. And you can see already there actually is some pollen stuck to it. So these have pollen stuck to it. Now you might be thinking to yourself, oh hey, this plant is doing a terrible job of reproducing because it's producing pollen and the stigma is right next to where the pollen sacs are on the anther, and it's going to fertilize itself. True. So plants want to avoid this if possible, because this is basically clonal reproduction. And remember that the entire point of doing sexual reproduction is to increase diversity. If you fertilize yourself, you have not increased diversity. So self-fertilization is usually detrimental for sexually reproducing plants. They don't do it intentionally, and they usually have little tricks that they use in order to prevent them from fertilizing themselves. So there's a couple different ways they can do it. We're not going to talk about all the different strategies that are at work here, but a lot of them are things like when the pollen is being released from the pollen sacs, at that time, the stigma, which is at the top here, the sticky part, this part right here, is not sticky. So it won't produce whatever protein makes it sticky at that point. And then later on, when its own pollen sacs become active, when its pollen sacs are no longer active, when they are done producing their pollen and pollinators have carried them away, then their stigma will get sticky. So that's one strategy which can prevent, or at least help prevent, self-pollination. You're going to get some self-pollinating no matter what. Some plants are almost completely reliant on self-pollinating, like dandelions. I didn't realize this, but they mostly self-pollinate. So even though they have all the structures for sexual reproduction, they don't really use them for sexual reproduction. They mostly self-pollinate, which I found fascinating. I actually learned that from one of the students' projects a student did a project on dandelions, and I'd never heard of that before. And I thought, well, that's got to be wrong. There's no way that it has all the organs for sexual reproduction. It's got filaments and anthers and stigmas and styles, but I looked it up, and it is true. I was shocked. So it's set up to do sexual reproduction, but most of the time it actually clonally reproduces, which is kind of cool. Anyway, so we've got, like I said, stigma at the top, then the style is right here, and then at the bottom, and this is going to be impossible for me to show you this. If I move the microscope around, it's going to get all blurry, but at the bottom, there'll be sort of like a little bulbous area, and within that, that's the ovary at the bottom of this thing, and you're going to find an ovule inside. Now, I'm fairly certain for buttercups, each one of their ovaries only has one single ovule in it. And so that ovule, if you cut this open, you can actually see the ovule, the egg cell. And so that is what's going to bind together. So what the pollen actually does is it'll grow a little tiny tube that goes down here. And the tube will grow right in to the ovule. And then the pollen grain will actually send its nucleus, which is one N, down the tube and in. So it grows like it's called a pollen tube. It grows a little tube in, and then it's going to allow its DNA to merge with the DNA in the ovule. Plants have different strategies for this. Ovules usually often have more than one nucleus. So this is a little bit complicated. We're going to talk about it more tomorrow. But one of those nuclei is one N, and that is what's going to merge with what's in the pollen, and that is what makes the baby, the zygote for this plant. The other nuclei inside is often 2N, and it's 1N, and it'll join with a separate pollen, and it's going to make a material. It doesn't make a baby plant. It only produces a material that the zygote can feed off of at the beginning of its growth. And often that makes up the bulk of the material inside the seed. Again, we're going to talk about this more tomorrow, but those are the basic structures. So we've got a stigma here, a style, and then at the bottom there's going to be like a little bulbous part that's going to contain the actual ovule. Okay, so that's what it looks like for buttercups. This is one way to configure these sexual organs on a flower. Let's look at a different one for comparison's sake. Okay, so I'm going to switch my lens here to something a little bit smaller. And so let's get a macro view first of this second one, and I'm going to. I think my iPad disconnected, of course. I don't want to take up too much time. I realize that I'm being long-winded here because it's so difficult to get this stuff into focus. But let me show you a different configuration at least, so you can see something a little bit different. Come on. I won't go through all these because they're all very similar. How's this working? Oh shoot! iPad, drive me nuts with this stuff. I have to set up a separate Wi-Fi network that is just for it's like a hotspot basically, in order to get this connection to work reliably. I don't know why. Of course, now I can't find the network. Why can't I find it? There we go. All right. So what I'm going to do here is show you this flower as an alternative. It's a little bit larger too, which will make it quite a bit easier to find these structures. But I'm going to have to cut it in half in order to see what's below. So if you look at the top here, you'll notice that you can't actually see, well, you can't see much. It looks like the anther and the filament and the stigma and the style are all hidden inside. And this is intentional because this flower has a really cool reproductive strategy in that it is specific to only a couple different pollinators. Only specific pollinators can actually get in here in order to properly pollinate this thing. These particular flowers do not like bees, and they are pollinated mostly by things like hummingbirds and hummingbird moths, which have a very similar proboscis. And there are some other insect pollinators that have a long proboscis, and the proboscis is just like basically like a beak. A hummingbird has a beak, and insects have a proboscis. So if you look, most of the structures are hidden down in this little area at the bottom. And so you need a long proboscis or a long beak in order to actually get down into the bottom part there. And the reason that they are specific to a specific pollinator is really for efficiency. So when flowers lure in pollinators, they're offering something to the pollinator. Food, basically. Either nectar, or the pollinators are using the pollen itself as a food source. And so if you have just a specific pollinator that can get into your flower, it makes it much more efficient. You basically only have to feed the pollinators that can get in there. And you know that those pollinators are going to prefer your flowers. So when they go to other places, they're mostly just going to be going to other flowers that are your flower, the same species or similar species. So that just makes the process a lot more efficient. So in other words, a pollinator and a flower or a plant have co-evolved. They've evolved to essentially mesh with each other very well in their reproduction strategy. There's many examples of this in nature. There's a bunch of really cool examples of this in the video. So in order to take a look at the internal structures here, I've actually got to cut it open. I'm going to use a pair of scissors. I wish I had a dissection kit here with a scalpel and some, but I don't. So I'm just going to have to make do with a pair of scissors, which hopefully isn't going to butcher this thing hideously. I'm hoping it won't. And then we can take a look at some of these structures. Okay, here we go. Here we go. Oh, I cut off a bunch of them. Whoops-a-daisy. I'm going to pull the petals off because they're just in my way. But let's take a look at the stigma and the styles. Oh, and also the anthers and the filaments. So I'm going to put these under my microscope. Let's zoom right in. I'm going to sweep all this stuff from the other flower away here. And I'm going to see if I can give you guys a really good view of these. All right, here we go. Oh, I think I found it. That looks right. Why did it just do that? No. Oh, that's petal. That's petal. That's not what we want. I'm looking in the wrong spot. Oh, looks promising. Petal. Now you might notice that the petals have this like really Ooh, I just saw something. I think that was it right there. Petals have this iridescent quality when you look at them under the microscope. That's actually because they're reflecting a bunch of ultraviolet light, which is something that is very appetizing to a lot of insect pollinators. Oh. Well, there's a filament that's going to have an anther at the end. If I can find the thing. That yellow that you see there is it. Come on. Oh. Come on. Man, is this stuff ever difficult to get into view? I mean, it's a three-dimensional structure, so it can be a real pain. Now it's stuck to the microscope. Fantastic. I promise I'm doing my best here. There is one right there. Oh, shoot. There. There. So what we're looking at right here is a anther. It's not in the best focus, but these little bumps right here are grains of pollen. Grain of pollen, grain of pollen. Okay, let's see if we can get one of the female parts here. The female part. Oh, I think I massacred this poor thing. I can't even see the female parts. Well, I think I destroyed this one. Okay, hold on. Let me open another one. I don't even see the female parts inside it anymore. This one only has a single stigma in the middle of it. For the life of me, I cannot find the thing. Where is it? See a bunch of anthers. Oh. Now, it's possible. So some plants also separate their male and female parts. So some flowers will have the stigma and style and ovary, and some flowers will have the filaments and anthers. And so actually it's possible that this is that type of flower. It'd be helpful if I had a real hardcore botanist here with me to help with this, because I don't really know a lot about local flora here. I'm not finding a stigma in the ones that I've opened so far, so I'm wondering if these ones only have the male parts. Okay, here we go. Here we go. This one's got some female parts in here. So this one actually has multiple. Okay, so it's got a bunch of filaments and anthers, and then at the base there are several stigmas, styles, and ovaries. That structure, by the way, altogether, stigma, style, and ovary, is called a carpel. I don't think I'm not. I should use the terminology. Let me see if I can get a good view of a carpel. It was certainly much more obvious on the last one. I might have to go down a magnification. This might be too strong a magnification. I'm having a lot of difficulty getting this shot. So what you're looking at there, that's just petal. You can still see the cells of the petal, which is kind of cool, but this is not being helpful. Okay, let me try the other magnification real quick, and then I'm going to let you guys watch the documentary and move on. I'm not trying to tie up your entire day here. This microscope is a little bit finicky. Oh, perfect. Oh, I should just use this magnification from the start. You can see stuff way better here. So that, why didn't I just use this magnification from the start? So here, these are the male parts. We've got anther, and then filament holding the anther. Now, where is the female part? I'm not able to find it under this thing. Anyway, they're there. Actually, I might be looking at it right there. There we go. There we go. There we go. Stop moving. There we go. Stigma. Right here. And then I'm not going to be able to move to this thing, but here's the stigma. And this is the style, and then at the bottom is where you would find the ovary with the ovule in it. I think each one of these has just one single ovule. The ovary, which is the bulbous part at the bottom of each carpel, is what becomes the fruit. So if there are multiple of these, you're going to get multiple fruits per flower. If there is one single one of these, you're going to get one fruit per flower. So an apple, if we look at an apple flower, you'll see that it only has one carpel in it, and that's because each apple flower is going to form one apple. Can the flowers look different for male and female? Yes, they can. They are sometimes completely structurally different from each other, which is kind of cool. I don't know about petal difference. That I'm not sure about, because number of petals is usually attached to whether or not they're a monocot or a dicot. That is a great question, Lily. No, I'm not going to look that one up. That I'm not sure about. I think the male and female have the same number of petals. Well, one way to find out. I'll look that up. I'm not finding an obvious fast answer to this, Lillian. I am not 100% sure. Certainly there are plants that have imperfect flowers that have male or female parts, and the flowers do look different. I don't know about petal count, though. That's fascinating. You know what you could do? you could just have a look inside those flowers. If one of them only has the female parts and one of them only has the male, then you got your answer. A little field research. Anyway, I think that's as deep as I'm going to go on this flower dissection. I could do this with each one, but I think it'll take forever, and you probably don't want to sit around while I do that. So what I'm going to do then is just let you guys move on. You're going to fill in the table that is right underneath that flower diagram in your notes, and it's just basically the basic function of each of these parts, which you should know. And you can also use page 598 in the textbook. That's where you find that information as well. You watch the documentary, and then there are those 2 questions to consider. I'd read the questions first because it's what you're supposed to be thinking about while you watch the documentary about the evolution of flowers. It's a fascinating documentary. There are 2 homework questions, 7 and 8 on page 602, and then a quick little exit quiz. I will not be available during Learning Block 3 today. I got to pop by the school to pick up apparently a new Chromebook. So I'm going to exchange my old Chromebook for a new Chromebook. I don't know exactly what's going on with that, but I won't be here during Learning Block 3. But please still send emails. Okay, I won't be like on the meet, but I will be responding to your emails. I'll get to them as soon as I can. So, but anyway, that's what we're going to do today. A little bit different than on a normal day. Okay, guys, I'll let you get to it. Enjoy the documentary. It is fascinating. There's 2 other parts, by the way. If you can find them online, they're well worth a watch as well. All right. Let me know if you need anything.